relatively persistent traits and habits. Just individuality, if you like. And I'm sure that everyone in this room probably finds questions about the origins of these differences among people intrinsically interesting. But from a biomedical perspective, the incentive to study them comes from the well-documented observation that many seriously adverse outcomes evolve disproportionately in the context of specific behavioral phenotypes. And among these adverse outcomes are substance use disorders and affect disorders and even psychotic disorders. And more prevalent things like academic failure, ADHD, dyslexia, dyscalculia, different kinds of social dysfunction, incarceration. So from the time of conception to the onset or the diagnosis of these adverse outcomes, the evolving intermediate phenotypes are really just different forms of individuality. So what do we know now that constrains our models of the origins of these differences? Well, we have known for a while from twin and other family studies that many of these personal attributes exhibit moderate variability. And some studies have identified polymorphisms that may mark causal variance in the link between genetic variation and these personal attributes. However, a combination of GWAS results and estimates of the heritability attributable to common genetic variation in aggregate, so-called chip variability, suggests that while much of this variability may be explained by common variants, the genetic architecture of these traits is likely to be highly polygenic. But whatever the origins, whether they be genetic or otherwise, of one thing we can be sure. Most behavioral phenotypes do not emerge at a particular time, but gradually, through the cumulative effects of factors that influence them over the life of the individual. For example, the left-hand figure here shows the long time course of developing calculation skills in children. And surprisingly, even the basic capacity to inhibit a primed motor response, shown here on the right, exhibits a similarly protracted course of development. But why do behavioral phenotypes appear to diverge during development? Why do some children from this study at age 14 exhibit calculation skills more typical of the average 9-year-old? And some children at 9 exhibit motor inhibitory functions more typical of 15-year-olds. Again, there's evidence for some degree of genetic mediation of the variability on both of these kinds of constructs. But developing calculation skills are obviously strongly modified by the instruction and learning experiences of children and by other cultural factors. And even performance on a simple motor inhibitory task like this one can be shown to be substantially modified by practice. So in our group, we begin with this basic conceptual model for an emerging behavioral phenotype. We can think of it as the function of, first, a hypothetical domain-relevant neural genotype. And in some circumstances, environmental effects on the neural apparatus relevant to the skill or domain. For example, as might occur in early brain damage or toxicity or other effects, including epigenetic effects. But more generally, the cumulative experiences of the individual encountering and manipulating material in the domain, and what are likely to be important interactions between these factors. In this context, I think it behooves us as developmental scientists to keep sight of a couple of what I call big questions. Now, these are obviously my favorites, but I think that in some form, these questions strike at the heart of matters that must be resolved if we are ever to achieve our translational aims, and that is to prevent adverse outcomes and lift the trajectories of well-being in at-risk individuals in our society. So here's my big question one. To what degree and via which biological mechanisms Does common genetic variation constrain or bias the functions of maturing neural circuits in the human brain? And here's my big question number 2, probably 2. To what degree and under which circumstances are there consequences of these genetic effects for experience-dependent developmental processes, or for the responses of the developing brain to neuroactive environmental factors such as trauma or toxicity or drug and alcohol exposure and things like that? So we might ask the question, can observing the biological development of the brain in children help answer these questions? Now that seems like a silly question to ask in this company, but until not too long ago, I can assure you that it seemed to many people to be quite unlikely. In part because the time course of the evolution of these complex behavioral phenotypes seemed much more protracted than what was thought of as a foreshortened period of the brain's biological development. And thus it was assumed by many that although early events in development might fix the range of outcomes, everything happening in the school-age years and beyond was necessarily due to learning in a biologically mature brain. And this, of course, changed quite dramatically with the advent of non-invasive imaging. So now I'm going to ask you to indulge me while I revisit one of the first papers I published in this area with my UCSD colleagues 23 years ago. Not certainly because it is a particularly interesting paper. On the contrary, it will best serve to illustrate how far we have come in the last 20 years. But I'm pretty sure it is one of the earliest imaging papers to cite Peter Huttenlocher's remarkable work. Now, I'm going to take some credit for, as Roger said, for attempting to develop some of the first semi-automated morphometry methods for use with MRI, and for insisting on the importance of applying stereotactic methods for achieving better anatomical registration, which, believe it or not, people did not do at that time. But nevertheless, these ROIs, which are essentially quadrants of the cortex, illustrate that we are going very far back from our elegant surface-based methods that we apply today in this study. Nevertheless, we did use these ROIs and these methods to measure cortical gray matter volumes after performing tissue segmentation based on dual echo MR images. And in this fairly modest sample of 39 young people ranging in age from 7 to 35, we observed highly significant decreases in our cortical gray matter volumes in these two dorsally lying regions, and really we found actually no evidence for any age-related reduction in the cortical volume in these more ventrally lying regions. Now, obviously, we know a lot more now about the age functions and anatomical pattern of these apparent changes in gray matter volume, but at the time they were quite surprising to many people. And the biological explanation was far from clear. Since the one largely postnatal phenomenon of which we were all aware was the protracted course of myelination, we and everyone else suggested that myelination, either of axons coursing just beneath the cortical rim or even intracortically, might contribute to these apparent volume reductions. But we had also just become aware of Huttenlocher's very interesting observations of synaptic pruning and what might be an anatomically heterochronous pattern, which was kind of what we were seeing, this heterochronous change. And so we speculated that the processes that we were observing might be related in some way to the processes that led to synaptic pruning as Huttenlocher had observed. Now, before I move into the modern era of neuroimaging, I'd like to point out one result in this paper that virtually never gets repeated in the subsequent literature, which was actually an important reason for me for continuing to seek explanations for our findings that included both true regression of tissue volume as well as change in contrast, as might be expected from ongoing myelination. And that is the observation that we made in a very similar pattern to the cortical volume reductions. We also saw a significant increase in the cerebrospinal fluid volumes in the adjacent regions of the subarachnoid space, as though nearby tissue process of ex vacuo expansion in this space. So this is something people never talk about, but which certainly influenced my thinking at the time. Well, obviously, since these early observations, much more elegant work has been done, most of it by all of you, very little of it by me, demonstrating that there are many quite robust indices of the ongoing biological development of the brain that can be monitored non-invasively in children, and that many of these show very protracted developmental trajectories of change that vary in form and a complex pattern from region to region. And I'm not going to go through all of these. This is just because it's hardly an exhaustive list, but you can just see where there's a constantly growing sort of array of signals that track biological development of the brain and functional development. But I would like to illustrate a couple of these effects using data from a project that we call PING, on which several of us here today collaborated, and which was funded by NIDA and NICHD as an RC2 mechanism, one of those from the stimulus package, basically. And this study, as you can see, first of all, it involved ultimately 10 data collection sites, and it was really a dream team of leading developmental cognitive neuroscientists and pediatric neuroimaging at many centers throughout the country. We had only one specific aim, which was to create a public pediatric imaging genomics database, which had this scope that was this large and that covered this entire age range from 3 to 20. The study was entirely cross-sectional and unfortunately involved only a very limited number of phenotypes, and they're listed here. We did have multimodal imaging and genome phenotyping, of course. But the data set is now available freely to the research community, and it's been accessed by people from all over the world through a web-based tool called the PING portal that we developed and that we continue to support for download and exploration of the data. And I'm just going to show you a couple of the, I'm going to show you some data that sort of illustrates some screenshots from the data exploration portal. For two of our primary global cortical phenotypes, this is total cortical surface area up here, and this is mean cortical thickness down here. I just want to illustrate one thing that's important in exploring developmental data like this. You have to have the utility for modeling highly nonlinear and often non-monotonic functions, because these age functions are very idiosyncratic. And what you'll see here is that there's evidence for subtle but highly statistically significant increase in cortical surface area up until this kind of prepubertal region here, area here, and then even subtler but again highly statistically significant contraction in total surface area. Thereafter, we see a very different pattern in mean cortical thickness, which is essentially linear across this entire age range. But now I'm going to drill down a little bit on this effect up here in the surface area of the cortex, and I'll show you another feature of the portal, which allows us to estimate effects of cortical phenotypes at the vertex level and create visualizations like these, which code for each vertex, in this case, an age-specific estimate of the instantaneous annualized rate of change. And I'm going to try to, it's a little bit difficult to have one figure that sort of shows this kind of a complex pattern, but what we're mapping here at each vertex shows that at ages 3 and 5, we estimate that at essentially every point on the surface of the cortex, the surface is expanding in excess of 2% a year. By age 7, you can see that in some regions that expansion is decelerating, but in some regions, limited regions, it's still expanding in excess of 2% a year. By age 9, there's very little expansion or contraction, but you can begin to see that there's some evidence of contraction. By age 11, the areas showing contraction are more definite, and afterwards you see that contraction of the surface area of the cortex sort of proceeds throughout the entire cortex. Now, these findings describe, from a cross-sectional data set, the modal course of surface area expansion and contraction across development. But you might ask, what do we know about individual differences differences in cortical areal expansion or cortical regionalization? Well, we know from rodent models that arealization of the cortex is linked to gradients of secreted morphogens and the expression of transcription factors in the neocortical proliferative zone during the embryonic period. And transgenic models have shown that alterations in these gradients lead to different relative sizes of the primary sensorimotor regions in the later developing cortex. And so an imaging genomics group in our institution, with whom I work, reasoned that genetic variation might give rise to individual differences in human cortical regionalization. And so, using data from a group of 400 middle-aged male veterans from the Vietnam-era twin study cohort, Chihua Chen, working with Bill Brennan's and Anders Bale's groups, applied conventional twin methodology to compute the genetic correlation of a measure of cortical surface expansion at each cortical vertex location. There were 2,300 vertices after smoothing, with the measure at every other vertex. So this obviously generates a large matrix of genetic correlations, vertex to vertex correlations. And then this genetic correlation matrix was further analyzed with a data-driven fuzzy clustering technique to identify 12 sets or clusters of vertices with relatively higher genetic intercorrelations and relatively lower genetic correlations with other sets or clusters of vertices. And these clusters are color-coded in this bottom row. This is a pattern, these are results that were reported in Science by Chihua Chen and colleagues in 2012. And what these show is basically that individual differences in the relative size of, say, this region in the dorsomedial prefrontal cortex these individual differences are heritable, and the genetic factors that seem to contribute to that individual difference variability are relatively distinct from the genetic factors that contribute to individual difference variability in the relative size of, say, the occipital cortex or the ventromedial prefrontal cortex, and so on. And so this tells us something about the degree to which surface arealization or cortical regionalization in human beings is genetically complex, and that is to say that it is very genetically complex. And it also is a nice way of sort of coming up with a novel parcellation of the cortex, which, if you will, is sort of genetically informed. And so we have exploited that in some of our further studies. So this was one of the first papers that came out of King. It was written up by Anders Bale, who was doing sabbatical leave in Anders Bale's laboratory at the time. And he looked at this question about whether individual differences in cortical areal expansion might be related to behavioral phenotypes in the King cohort. And when he looked at this sort of classic incongruity effect using the flanker task, just looking at the comparison between the speed with which kids could identify the direction of this central fissure in the incongruent versus the congruent condition. He looked at a subset of the subjects. You can see that this is a function which improves dramatically across the age range. This is just showing improving performance on the flanker task. And what he found was that in a region that was primarily in the anterior cingulate, relative cortical expansion in this region was associated with stronger performance on this measure, controlling for age, gender, SES, genetic ancestry, scanner, and other things. In another study that was done by Eric Goodman, who's sitting right down here, who's a member of the Center for Human Development group, we looked at some data that we had in King in a subset of the individuals who were a little older, the kids who were 8 and up, on a measure of generalized anxiety. And we were particularly interested, again, we used this sort of genetic parcellation, and we were particularly interested in whether this region shown in pink, which seemed to us to be a very good delineation of the ventromedial prefrontal cortex, which has been strongly implicated in emotion processing and anxiety in particular in other contexts. We were interested in whether there was a difference in the degree of cortical arealization or surface area expansion in children who were self-reporting high levels of anxiety versus low levels of anxiety. Indeed, there was. But particularly interesting to us was the fact that there was an interaction with age. So it appeared that possibly children who experience high anxiety show a somewhat later expansion of this region relative to kids who have lower anxiety. And there's a lot more to this story, and Eric will be telling that story in this poster, so I would encourage anyone who's interested to go and hear about it. There are more interesting pictures to see, and also there's more to the story having to do with fitness phenotypes. I'm just going to close with this observation from Ping to say that in other contexts, these are data actually from combining our studies in UCSD with studies in Denmark. We were focusing on these two phenotypes. This one is the classical stop signal reaction time, the measure I showed you earlier of sort of motor response inhibition. And this one is a measure of perceptual decision making, which involves the detection of motion coherence. In both cases, you see there's very strong improvement in performance across this age range from 6 to 20. And in both cases, we were able to see an association with the polymorphism in the dopamine this is a VNTR polymorphism in the dopamine transporter gene, such that the 9R carriers genotype seem to show a distinct developmental trajectory on this measure relative to the 10R homozygotes. So again, in these last two examples, what I'm trying to highlight is that in some cases what we're seeing is that either the trajectory in a neural phenotype or the trajectory in a behavioral phenotype is what is best mapping on to the other domain. So these observations really are just typical of many made in recent years by those of you studying brain and behavioral development with imaging. And I think this is a fair general summary of them. First of all, I think it's obvious that biological and functional development of the brain can be monitored in vivo. And behavioral phenotypic variability in developing children is mirrored to some degree in the imaging variability. In some cases, this correspondence can be observed at the level of trajectories, like in the last two cases that I showed you. And both behavioral and imaging phenotypes do appear to be mediated to some degree by common genetic variation. But what do these associations really mean? Are the associations genetically mediated? And if they are, does this represent differing functional constraints within the circuitry of different individuals? Or does it represent the effects of different functional biases rather than constraints per se? In either case, when does such constraint or bias emerge during the functional development of the relevant neural systems? Unfortunately, we don't have a clear understanding of the meaning of these associations in these terms. And partly for this reason, nor do they point clearly toward any particularly promising avenues for intervention. And a significant problem is that we don't yet know what the imaging differences represent at a cellular level, and thus how to use them to help us answer those big questions that I posed earlier. For example, if we just focus on the developmental signals that we can monitor in maturing human brains, what differences in the structure of the cortex attend early cortical surface expansion and later contraction? Is it differences in columnar structure, dendritic morphology? Is it due to maturing neuronal or glial phenotypes? Is it ongoing proliferation and differentiation in some glial or even microglial populations? What about the alterations that we measure as apparent cortical thinning? Is it associated with changes in dendritic mass or morphology, laminar architecture, intracortical or subcortical myelination? We still don't know. And what about the cellular correlates of these remarkable changes in diffusion parameters that we're so interested in? To what extent are we measuring myelination? Because there's plenty of evidence in many cases it's not what we're measuring. Is it neurite diameter or tortuosity? Is it some shift in the extracellular versus the intracellular signal, cell density, or in many other possibilities? And we still don't know much about what's driving these changes either. We don't know what the cellular changes are dependent, and we don't know what's driving them. What are the associated changes in gene expression? What is the role of experience in neural activity and epigenetic modifications? We need to know more about what neural factors mediate the associations that we see between behavioral and neural phenotypes. Are they genetically mediated differences in neural architecture? Or could they just be genetically mediated differences in the time course of brain development in otherwise similar neural architectures? What are the effects of practice, training, or experienced emotion via activity-dependent processes in the relevant circuits? Are they mediated by gene experience correlations and interactions? So I'm arguing today that in order to answer these questions, we need to take the research to a different level. And that a significant impediment to that aim is an unfortunate degree of fragmentation in developmental science generally, and in the science of the developing human mind and brain in particular. And I point to these attributes of our field. Human developmental neuroscience, developmental psychology, child psychiatry, pediatric neurology, educational research, and many other relevant branches of developmental science remain siloed with few unifying or integrative structures. And perhaps in part because of this, the support for our research is provided through many small programs distributed across multiple institutes of the National Institutes of Health and multiple agencies outside of the National Institutes of Health and other foundations. And our discipline of human developmental neuroscience is even further isolated from basic developmental neurobiology, even though both of these disciplines have much to gain from closer integration. So how do we achieve the integration that we need to answer these big questions? Well, I think we have to start at home, and we have to start by actually observing the developing mind closely and continuously. And I say this because I don't think we've actually adequately done this yet. We certainly haven't done it with the toolkit of modern biomedical techniques that we can bring to bear on it today. We need to do it by beginning to accumulate large, informative, high-dimensional data sets for sharing, in my opinion. Populated with observations from developing children obtained repeatedly over time as they mature. And in all cases possible, we need to collect comprehensive genetic and epigenetic variables. Of course, we have to apply non-invasive imaging and other neurophysiological measures that are sensitive to differences in neural architecture, the status of the brain's biological development, and activity within maturing biological systems. This is critical. We must collect a much denser corpus of behavioral and experiential assessments, and we should use our conventional methods like formal testing, observation, and parent self-report, but increasingly we should use behavior sampling with mobile devices and other modern data collection techniques. We probably are going to get lots of interesting information from new generations of so-called quantified life apps. We can collect very high-dimensional information about physiology and activity. And there are many other interesting behaviors. And while we're observing the developing mind, and if we're going to observe the developing mind in this way, I think we have to recognize that we're going to have to establish entirely new relationships with families. Because what we're talking about here is a very long-term partnership with families who are going to be willing to allow us to observe their children closely over a long period of time. And so I think we're going to have to recognize that these new relationships with families have got to clearly add value to their lives. It's unrealistic to expect them to have this kind of partnership with us over a long period if we don't find ways to enrich their lives through these experiences. And so I think we're going to have to get very creative in learning how to do this. Another thing I think is extremely important is whenever opportunities exist, we should embed translational arms within large-scale longitudinal studies. This would lead to prototyping, testing, and modification of novel interventions for mitigating risks in the struggling or the higher risk subgroups of our cohorts, as well as interventions for otherwise enhancing learning and growth within these high information cohorts. And thus the additional behavioral, developmental, neurobiological, and genetic information that would already be available for these participants can be leveraged in models of the effects or the lack of effects of these interventions. And the availability of elective interventions, for example, after-school or summer programs held in the same venues as ongoing longitudinal studies, or perhaps via web-based interactive applications, could also help to consolidate longer-term relationships with some families in our cohorts, and would serve as a catalyst for more interdisciplinary work on translation, for example, involving teams of teachers and clinicians as well as developmental scientists. So finally, I think a more integrated science would involve nested basic science programs explicitly designed to bridge gaps between the results from non-invasive studies of children and those of parallel studies of relevant cellular and molecular phenomena in animals and in human postmortem children. Interdisciplinary teams can propose new studies designed collaboratively by scientists who are studying human children and scientists who are studying animal models that attempt to create new bridging behavioral paradigms for cross-species comparisons, or that examine animals with non-invasive methods parallel to those used in human children to help define the neurobiological sources of these robust developmental signals that we can now obtain. So I'm going to start wrapping up now by summarizing my ideas about how a more integrated framework might work. And I'm going to start with what I think we, as the extramural research community, need to do. We need to together develop rubrics for harmonization of our methods, when in so doing, we would incur no cost to the quality of our science. And I think there are many opportunities to do that. As I said, I think we should stretch out and try to embed translational components into our observational studies. We need to move toward early data release and new standards for replication and secondary analysis that would be appropriate when we have larger shared data resources to use for that purpose. We need to move to create nested multi-scale programs and prioritize transdisciplinary collaborative efforts. Again, we have to try to design links for bridging the gaps. These are a few of the ways we might try to do that. Another thing I think we have to attend to, this is for cognitive neuroscience generally, but for us too, we have to figure out how to create public repositories for multi-scale data, because even some of the best public repositories we have now tend to focus on data at a particular level of analysis, and ultimately we need to have multi-scale repositories, and we need to figure out how to do that. So this is what we need to do. But there are some things that we need to get. There are some things that we need to have our sponsors do for us. And some of them are that we need structures for integrating and leveraging resources from across the institutes within the NIH and broadly with the NSF, Department of Education, and other sources. And with those new integrated resources, we need mechanisms for funding of multiple PI, multidisciplinary collaborations that go across level, across species, and that, as I say, embed interventions and observational studies. We also need support for infrastructure and informatics specifically designed for developmental data. Because it turns out that some of the large informatics initiatives that are really focused on adult data don't really work that well. Some of us have found out the hard way when you try to apply them to do development studies for a number of reasons. We want the institutes, I think, to adjust the review criteria to incentivize this kind of research. I think that you should than you got from your outstanding science, if in addition to your outstanding science, you attempted to harmonize your methods to some degree so that you can contribute more to sharing the repository. So I think you need to get points for that. I also think that we have to move beyond just requiring people to have data sharing plans to evaluating them at renewal time with respect to whether they did in fact release and share their data. And I think that we need, in the same way, perhaps not to the same degree, but in the same way that we explicitly give people credit for innovation in their research, we should give them some credit for integrative elements, because after all, those are high-risk things to do, to reach out and try something risky that integrates across cycles. Okay, so in conclusion, I'd like to say that the remarkable progress that has been made in developmental cognitive neuroscience in the last 2 decades is undeniable and very exciting. But I fear that progress in answering those big questions about the human mind will languish unless we demand and create a more integrated science of the developing mind and brain. I would be the first to admit that the most daunting challenge that we face is the enormous complexity of the phenomenon of interest. But in my opinion, this challenge is all the greater when we are hamstrung by an unnecessary degree of fragmentation in our field. You know, no societal campaign is more universally embraced than the pledge to ensure that each child can reach his or her own potential. But there is no consensus about how to accomplish this. And this situation reflects a sobering fact. Insufficient evidence exists to reconcile contradictory assumptions and predictions. The answers to our questions are not beyond our reach, but they may be coming a little too slowly because we are not deploying our scientific assets in the most powerful way to answer our big questions. And I think we should change that. Obviously, I'm just a minor player in a team of all-stars, and this includes the Ping investigators, my colleagues at the Center for New Development, the Vets and Emerging Genomics Group. I want to especially acknowledge Chi-Hua Chen, NICHD, and NIDA, obviously, for funding. These are just some of the people whose work I highlighted here. Tim Brown's beautiful annualized rate of change pictures, and go listen to him give a very interesting story about the importance of the structure of Ping and the genetic ancestry factor. Eric Newman, as I mentioned, in the anxiety work. I want to acknowledge Hauke Bartsch and Wesley Thompson, who made critical contributions to the development of this data exploration and modeling modules in the Ping portal. And last but certainly not least, my good buddy Anders Dale, without whose good offices I wouldn't have very little to talk to you about today. So, I'm sorry, I had to make this.
So we have time for a couple of questions if you're willing to answer them.
You know, she asked for questions, but I was actually hoping for comments as well, because you know, I know that I don't think I suggested is probably seems like great, maybe not, and I'd be interested in what people think about this.
So can you provide an example of how you do this integrated approach? So you're thinking, in an institute, start talking to our animal model colleagues, try to bring in the post-mortem people who are so far away in flux, is really trying to bridge this gap.
Yes, I can see that. It seems like a great idea. Well, I think, you know, I think the problem has been that, yes, all of those things need to happen. I think we need to reach out and find partners, willing collaborators, who will help us try to bridge the gap and try to, because, you know, there's very exciting stuff going on, as you know, in developmental neurobiology, but many times, you know, there's no obvious attempt even being made to bridge the gap. And sometimes, you know, on both sides, there are these kind of superficial references to what's going on the other side. And you can tell people are really not talking to each other. So I think we have to do that. We have to try to create venues for that. But it's kind of catch-22, and this is what I'm trying to refer to, because we all know that, you know, in order to do that, we need to have the hope of getting support to do that. There has to be a mechanism that would allow us to pursue those interesting ideas or pursue those interesting things. And I think this is the problem that I think is most critical. Because, you know, I spent, it's a little off topic, but I spent about 25 years doing HIV research. So I was really involved in neuro as part of the HIV story throughout, you know, from the beginning of the epidemic. And here, the situation was very different. The institutes took the position that all hands were on deck. Every institute had to contribute, and there were definitely integrated mechanisms for doing that. Every discipline had to be at every table, essentially. Every major age research center had to have people from all, I mean, people you could not imagine talking to each other before. You know, sexologists and virologists. And they were all at the table. And it was remarkably successful. And in the beginning, I can tell you that connecting the dots seemed impossible. So I guess I feel like this is possible, but there has to be both top-down and bottom-up action to come up with the ideas, but the ideas have to have a place to go. And so those of us in a position to talk to the leaders of institutes and policy leaders need to, I think, make them aware of the fact that this problem, understanding the developing mind and brain, is a critically important problem. That understanding it better could lift the trajectories of well-being for all generations of the future by preventing many adverse things from happening. This is a critically important problem. It's just as important as HIV. Not to say that HIV is not important, but it's solved. But, you know, it's at least as important as that because of its potential impact. And I think we have to keep explaining that it requires the same level of integration and urgency to some extent that some of these other visible problems.
Well, there are some representatives from different agencies here. I hope that they're listening. I mean, our study could really fulfill that. So, thank you so much. That was a very inspirational talk. I think we all needed to hear that. I mean, this is where we want to go to.
Well, I know that I'm preaching to the choir.
It is my honor to give you this plaque on behalf of Flux Society. So